terms of E1, almost everything I'm going to say right now is written here. But I would like to suggest that if you have just some blank paper, notebook paper, or anything like that, I would like to suggest that you pull that out. Section E deals with the subject, and I will admit it's not, quote, the sexiest subject in the world, but it's actually quite an important subject. Now, we're going to be using solutions in this class, various solutions, not a lot, but a little bit, and we should understand what is a solution. Now, in this case, we don't mean an answer to a test question, a math problem, but we mean a solution. A solution is made up of two things. A solution is made up of a solute, which we pour into a liquid. So we've got a beaker here that I drew, and whatever you add or dissolve in the liquid, that's called the solute. Now this solute could be salt, maybe we're pouring salt in the liquid, and the liquid's usually water. It doesn't have to be water. The liquid could be rubbing alcohol, it could be the alcohol that, like whiskey alcohol, it could be ammonia, it could be gasoline, it could be vegetable oil. But obviously, in biology and in medicine, the liquid usually is water. So whatever we add to the water, so if it's salt, it's a saltwater solution. If it's sugar, it's a sugar water solution. So a solution is made up of two things. If the liquid is water, we call it an aqueous solution. Aqueous. A-Q-U-E-O-U-S. It comes from the Latin aqua. Aqua, like aquatic, means water. And Spanish, being a Latin-based language, uses the word agua, so for water. All right, now, the next thing we want to address is, what is concentration? What do we mean by concentration of a solution? All right, so I drew two beakers, and in this first beaker, we're adding 1 spoon of, let's say, sugar to this a volume of water. And in this other beaker, we're adding 3 spoons of sugar to the same volume of water. So obviously, if we put 3 spoons of sugar in this water and only 1 spoon in this water, would everybody say that this has a higher concentration of sugar? Yes. Yeah. So what is the definition of concentration? Concentration is the amount of solute dissolved in a specific volume of liquid. It's the amount of solute, the amount of sugar, or the amount of salt, the amount of protein, the amount of whatever I've dissolved in a specific volume of liquid. That's called concentration. Everybody got that? No. No. How about now?
Yes.
Okay. So, now the next thing we want to look at is, what do we mean by percent concentration? There are different ways of quantifying, of numerically, using a number to express the concentration. In other words, we don't just say, we don't put 3 spoons of sugar in water, and we don't just say it's more concentrated. We should be able to quantify how concentrated is it. One of the ways that we express quantitatively, mathematically, it's not complicated, the actual concentration is called percent. Now, that percent sign should be very familiar. If you're a business major, you use percents in terms of interest rates and so on. In this case, the percent sign, percent or percentile, always means per 100. No matter how you use that percent sign, it means per 100. Percentile. Cent isn't 100 cents in a dollar? No, it's percent. So In this case, when we use the percent sign in terms of concentration of a substance in a solution, it's grams of solute per 100 milliliters of water. You can see we're now using metric system, right? So what's an example of this? So I've given you two examples. So we have something, we might run into something called a 5% glucose solution. Glucose is a sugar. Now I might just mention that another name for glucose is dextrose. Usually in biology classes we call it glucose. Interestingly, in hospitals they refer to it as dextrose. There's a reason why. Technically it's the D-isomer form of glucose, the dextro form. But so the 5% glucose. So what does 5% mean? Well, what did we say percent means? We said percent means grams per 100 milliliters. So can everybody see, I wrote in red, right, grams per 100 milliliters. That's where the percent sign was. And then what did I write here? 5 and glucose. Everybody see what I did? So literally, there's 5 glucose, and what's the percent mean? Grams per 100 milliliters. 5 grams of glucose per 100 milliliters of water. Now, you might say, why are we mentioning this? I mean, who cares? You're making my life miserable. This is the most common solution used in the hospital. Everybody see what it says? 5% dextrose. All right? And then another solution that's commonly used in the hospital, and the reason why I've got these is, I think many of you know, I teach physiology and pharmacology. That's actually what I do most of my time. I only teach one biology class, one biology lecture each semester, and I only teach one biology lab once a year. So I don't really teach biology much. But anyhow, another example is a 0.9% salt solution, NaCl. That's the second most common solution used in a hospital setting. Salt, when we use salt, we call it saline. How do you say salty in Spanish? Saline. Right. So it's called a 0.9% saline solution, or normal saline. And so I'm not giving you this number because I think it's hard. You know, it's simply the actual concentration of salt solution that's used clinically. I didn't bring the bags with me. I've got bags and solutions of all these things. So what's a 0.9% salt solution? 0.9 grams of salt per 100 milliliters of water. So we're just trying to explain what these mean. Because, again, this is an introduction to biology. So there's a few people in this class who are taking this class because they actually plan to take courses in anatomy and micro and physiology. Anybody here? Raise your hands. So there are people who are taking this to prepare them for those more prerequisite courses for the clinical fields. But no matter, even if you're not doing that, which most of you aren't, unfortunately I can predict with absolute 100% accuracy, unfortunately, all of us will end up in the hospital. All right? And you should know what's going on. And one of the things that makes hospitals and doctors scary is that most people have no clue as to what's going on. It's a very scary environment, and they don't explain a lot. And then when they explain it, most people don't understand what they just said. So part of what we're learning is to help us so that in the course of our lives, whether it's our own lives or the lives of our relatives, parents, our children, our friends, when we have to do with medical issues, we will have some knowledge about what they're talking about as doctors and nurses and pharmacists and so on. This is just a matter of time before you have to know what they're dealing with. And we've just explained a little bit what cancer is today. So all of this is important because it is part of the reality of life, is dealing with disease, illness, and eventually death. Right? So I hate to be morbid, but that's life. All right, so now the next thing we want to mention is what is diffusion? All right, now some of these topics you're going to hear more about in lecture as well. So what is diffusion? Diffusion is the name we give for the spontaneous movement of a solute from an area of higher concentration to an area of lower concentration. Yeah, if anybody needs paper, there's some more paper up here. Just have a piece of paper to write on. Again, it's all in the lab manual, but I want to just lay it out step by step. rather than just highlighting these words. All right, so what is diffusion? It's the spontaneous movement. You'd say, what's spontaneous? It just like happens on its own. The movement of a solute. You'd say, what's a solute? Sugar, salt, anything, oxygen. From an area of high concentration to an area of lower concentration. Or in other words, diffusion is when something moves down a concentration gradient from an area of higher to lower concentration. So what I have drawn here is I put a bunch of dots. You'd say, is that what you did? So these just represent either, these could be sugar molecules, these could be oxygen molecules, this could be any chemical substance. And what we see is that if we had a whole bunch of some chemical right here, it will spontaneously start to spread. It will do that on its own. And this spontaneous spreading out, as it moves from an area where there's a lot of it to an area where there's less, this outward spread is called diffusion. I'll give you a couple of quick examples. If I had a beaker of water and I dropped some blue dye into the water, we would see the blue dye start to spread or diffuse until eventually that blue dye would spread evenly, equally everywhere in the water. That's called diffusion. Another example, if I had a bottle of perfume, so there's a high concentration of perfume molecules in this perfume, in the bottle. If I take the cap off the bottle of perfume, what's the perfume going to spontaneously start to do? It's going to start to waft up and spread out of the bottle, isn't it? And soon we're going to start smelling that perfume in the air everywhere. Because that perfume is spontaneously, on its own, diffusing out of the bottle where there's a high concentration and spreading outwards throughout the air. That's called diffusion. It's a spontaneous process.
Does diffusion always go towards everything being in equal
Yes,
concentration?
yes. It will continue to spread until there's no longer a concentration gradient or concentration difference. So until that happens, it keeps spreading. So with that bottle of perfume, the more every single hour, you'll notice that smell is getting more intense as more and more perfume is spreading out of the bottle and spreading more and more into the air. Okay, so and it will continue basically until it's totally equalized the concentration everywhere. Now, let's do one more here. What does a cell membrane look like? Now, this is something that you'll be hearing about in a lecture, if you haven't already. What's a cell membrane look like? So, what I've drawn here, all right, so in case you're saying, what did you just, you moved that through that quickly. I wrote, what, number 5, what does the cell membrane look like? So, everybody got that? So, in our picture, so I drew a cell. This is the cell membrane, right? Here's the nucleus, right? Typical animal cell, could be a human sheet cell. And we've learned today that the cytoplasm, this jelly-like fluid on the inside of every cell, is about 80% water on the inside. And all living cells have to be surrounded by fluid. We have pointed out that if you're looking at any living cells, you've got to put water on the slide, otherwise it'll die. So all living things have to live in a fluid environment. And so this fluid on the outside of the cell is known as extracellular fluid. Extracellular means on the outside of the cell. Now in us, this extracellular fluid in humans is commonly called tissue fluid. So let me pose a couple of questions here. First off, on the surface of your skin, is your skin made up of cells?
Yes.
Yes, it's made up of cells. So you might say, but I don't feel any water here, Professor Fink. You said, don't cells, you said all living cells have to be surrounded by water. I did. Are these cells on the surface of your skin alive? No, they're all dead. And one of the reasons why they're all dead is because they're in contact with air. They can't continue living if they're exposed to air. If you've ever scraped your skin, which we all have, scraped your knee, scraped your elbow, scraped your arm, you've scraped off some of those dead cells. You ever see a clear fluid start to ooze? That means you've reached the layer of living cells. When you see that clear fluid oozing, that's that tissue fluid that's surrounding your living cells. When you open up somebody's abdomen and spread it open, there's all this fluid around all the organs. So all living cells have to be surrounded by fluid or they will die. And these are dead because they're in contact with air, not fluid. Now, the fluid really, in order to keep human cells alive, should also be the same concentration of water on the outside of the cells as on the inside. So if the inside of the cell is 80% water, the outside of the cells should similarly be 80% water. So now, in terms of what cell membranes look like, let me just pass out one thing to you. And this is something that the subject I'm covering right now, I can guarantee you with 100% certainty, just like 100% certainty I predicted, unfortunately, hospital issues, with 100% certainty in your lecture class, you will learn in much more detail what I'm about to cover right now. And you may have talked about cell membranes in Dr. Morrison's class yet? Yeah. So, yesterday? Good. So, okay. Now, I'm not going to get into as much detail as your lecture teacher will. So, what we're looking at is we are looking at the molecular structure of a cell membrane. So, this is a cell membrane. Right? This is the cell membrane. And the cell membrane is basically made up of two types of molecules, largely. It is made up of a double layer of what are called phospholipids. And I said double layer. They sometimes call that a bilayer of phospholipids. You'd say, does it say that somewhere? Right over here, it says bimolecular layer of lipid. Now, lipid means fat. So there's a double layer of fat molecules. Now, the type of fat this is called a phospholipid. Lipid means fat. It's a special type of fat called a phospholipid. And everybody knows lipid means fat because you've heard of liposuction. Now, there's a double layer. You'll notice there's an inner layer and an outer layer. Now, I'm just having fun coloring. Okay, there's a double layer of phospholipids. Now, each phospholipid kind of looks like, if I just kind of make an enlarged view of one of these guys, they kind of look like a balloon to me. They look kind of like a balloon with two strings. Everybody see that? That's kind of what a phospholipid looks like. All right? So each balloon, right, has two strings attached to it. It looks like this. It's called a phospholipid. Now, the balloon part, interestingly, is attracted to water. The balloon part of the phospholipid is attracted to water. We say it's hydrophilic. Hydro means, anybody know? Water. And philic means to like. So the balloon part likes water. The two strings, on the other hand, are hydrophobic. They hate water. Phobic means to hate. So we actually, every phospholipid molecule is schizophrenic. It's a schizophrenic molecule. Now, has everybody got that? Hydrophilic and hydrophobic. Now, you'll notice there, if this is the cell membrane, the cytoplasm on the inside of the cell is here. This is the cytoplasm. And the cytoplasm is 80% water. So this is this jelly-like fluid or water on the inside of the cell. And you will notice, therefore, that the balloon part, the part of the inner layer of phospholipids that likes water, the balloon part is facing the water on the inside of the cell. Now you'll notice there's an outer layer of phospholipids, isn't there? There's an outer layer, and they're upside down compared to the inner layer. So the balloon part of the outer layer of phospholipids is facing the tissue fluid on the outside of the cell. This would be the tissue fluid on the outside of the cell. Can everybody visualize that? So this double layer or bilayer of phospholipids, the balloon part that likes water always faces water, whether it's the water on the inside. Now, we've talked about diffusion earlier today. We said diffusion is this spontaneous movement of chemicals from an area where there's more of them to an area where there's less. Now we're asking the question, what are the 4 factors, obviously there must be 4, that affect the rate, how quickly these chemicals diffuse, specifically through a cell membrane, So the first factor that affects how quickly a chemical will diffuse in general, and specifically diffuse through a cell membrane, is temperature. Temperature, increasing heat, makes everything speed up. The warmer you make things, the faster atoms and molecules move. This is actually known as kinetic energy. So anytime you want to speed something up, you increase heat. Anybody who takes a regular chemistry course learns all about how the way you make any chemical reaction occur faster is you warm it up. And that just makes atoms and molecules vibrate and move faster and faster. Incidentally, if heat makes things move faster and faster, atoms and molecules move faster and faster, then cooling things down makes them move slower and slower. And if you're wondering, like, how cold would you have to make it to stop all movement of atoms and molecules? And you actually have to cool it down to a temperature known as absolute zero, which is actually minus 273 degrees centigrade. And when you cool things down to that temperature, which has been done, all atomic and molecular motion stops. That's called absolute zero. Anyhow, so temperature. We'll get to what the next thing is. So all that I indicated is an example of chemicals that diffuse across the cell membrane pretty easily are oxygen tends to diffuse in. Why? Because cells use oxygen. And in lecture, you either have learned or will be learning that cells use oxygen for something called cellular respiration. And in cellular respiration, as cells use up oxygen, they produce a waste product called CO2, carbon dioxide. So oxygen has to diffuse into the cell across that cell membrane, and carbon dioxide has to diffuse out of the cell across that cell membrane. The warmer it gets, the faster those chemicals zip in or out of the across that cell membrane. Just as an example of what I'm talking about, there are organisms that are said to be, the common expression is cold-blooded or warm-blooded. The better term is really poikilothermic or homeothermic, but we'll use those terms cold-blooded or warm-blooded. We are an example of a warm-blooded organism. How warm are we? Our body temperature normally is about 98.6 degrees Fahrenheit. That's almost 100 degrees. We're almost 100 degrees, our body temperature, which is a lot warmer than this air temperature in this room. Temperature in this room is probably about 70. So we're almost 30 degrees warmer than the air temperature. An example of an organism that's commonly said to be, quote, cold-blooded would be a snail. Okay? Its body temperature is not almost 100 degrees. All right? Think about how that affects the rate at which oxygen enters the cells of our body versus the rate at which oxygen enters the cells of a snail's body. You think that might have some effect as far as how quickly the chemical reactions occur and how fast it moves and everything else? So, and if we started cooling your body temperature down, you'd start moving and thinking, and everything else would be slowing down. And as the temperature rises, it starts to speed up. So that affects the rate at which chemicals go in and out of the cell and chemical reactions. Now, a second factor that affects the rate at which chemicals diffuse in or out of the cell is the difference in concentration between the inside and outside of the cell. We wrote that the bigger the difference in concentration, the faster the rate of diffusion. You'd say, what do you mean? So I drew pictures of two cells. You'd say, can you make it smaller so I can read it? Okay. All right, so I drew pictures of two cells. In both cases, I put a bunch of chemicals on the outside. In the top picture, there are none of that chemical on the inside. In the lower picture, there's already some of the chemical on the inside. So the question is, in which case, the top or the bottom example, where would the chemical move faster into that cell? The top. Because the bigger the difference in concentration, the faster the rate of diffusion. And when the difference in concentration is not as great, the rate at which it diffuses is slower. So the bigger the difference, and therefore, in the example I gave you earlier today of opening up a bottle of perfume, so initially, all the perfume molecules are in the bottle, and there are 0 perfume molecules in the air. So it tends to diffuse very rapidly. As more and more perfume molecules start to accumulate in the air, the rate at which these perfume molecules are diffusing out of the bottle of perfume will start to slow down. Does that make sense? So the rate of diffusion is related to the difference in the concentration gradient. Everybody all right on that? Yep. Now remember, did I put on my website various resources to help you understand this? Yes. The answer is yes. It's your choice whether you use them. Okay, now, we've given you 2 factors. A third factor. This is very simple. This is intuitive. Everybody will understand this. A third factor that affects how quickly a chemical moves across the cell membrane that diffuses is the size of the molecule. The smaller the molecule, the faster the rate of diffusion. So let's imagine 2 molecules, an oxygen and a glucose. Oxygen is 2 atoms big, O2. Even people who have me for a lecture, we've talked at least about an oxygen molecule. All right? Glucose is 24 atoms big, C6, H12, O6. It's got 24 atoms making up that molecule. All right? So I tried to draw them proportionally, that this one's smaller, that one's bigger. So which one gets across the cell membrane faster? That's obviously the small one does. In fact, if the molecule gets too big, it won't be able to get across the cell membrane at all. Obviously, when I drew these, in reality, molecules are not that big compared to a cell. But if I drew them relative to the size of the cell, they'd be too small for you to see either one of them. All right? And the fourth and last factor that affects the rate of diffusion. Whether the chemical... The next question that we want to take a look at is, what is osmosis? What is osmosis? Osmosis is simply, as I wrote, the diffusion of water either into the cell or out of the cell. Whenever water flows through that cell membrane, that's called osmosis. Why did we give a special name for osmosis, or for the diffusion of water, calling it osmosis? For all the other substances, we just call it diffusion. When oxygen diffuses into the cell, or carbon dioxide diffuses out of the cell down its concentration gradient. I guess the reason why a special name is given to when water diffuses is because water is really the only liquid that we commonly talk about, at least in a biological or medical sense. All the other substances that are flowing or diffusing into a cell or out of the cell are either solids or gases, and water is really the only liquid. So when this liquid water starts to flow in or out of a cell, we give it a special name, osmosis. Now, of course, water can flow either in or out, but if a whole lot of water were to start to flow into the cell that could cause the cell to swell up with water, even to the point where it might burst. On the other hand, if a whole lot of water were to flow out of the cell, then the entire cell would collapse. It would basically implode and collapse. That's actually, as we'll see, known as crenation. Now, the last topic that we want to address here is the question of tonicity. Now, you actually have a picture showing an analysis of tonicity on page E6 in the lab manual. This is actually page E6, and on E6 it shows what happens when a cell is placed in what's known as an isotonic solution, a hypotonic solution, or a hypertonic solution. You can look at these pictures, but let's take a look at a picture that I've drawn here, and we'll use this to help explain it. So what is tonicity? So in the first of these test tubes, it shows a cell. It happens to be a red blood cell, but it really doesn't matter. And it shows the red blood cell, where we are reminded that in all cells, the inside of a cell, the cytoplasm, is about 80% water and 20% other stuff by weight. And that other stuff would include proteins and sugars and vitamins and minerals and so on. So we've represented this other stuff, or solutes, by dots. Now, if that cell is placed in a solution that is similarly 80% water and 20% other stuff, other solutes, that solution would be called an isotonic solution. Iso is a Greek root that means same, equal, and tonic means strength. So it's the same strength, the same proportion of solutes and water in the fluid outside the cell as inside the cell. And obviously this is a happy cell because there's no concentration gradient for water to either rush into the cell or rush out of the cell. So the cell remains a constant volume. It neither swells up and bursts, nor does it collapse. Now, two examples of solutions commonly used in medicine that are isotonic are a 5% glucose solution and a 0.9% salt solution, commonly called a saline solution. Now, when I mention these as isotonic solutions, one question that might come to mind is, well, why aren't they the same number? Why is a 5% glucose solution isotonic and same with a 0.9% salt solution? So I want to remind you that a glucose molecule is a much larger molecule and weighs more than a salt molecule, which is much smaller and weighs less. So in other words, one molecule of glucose would weigh more than one molecule of salt. So in fact, 5 grams of glucose actually has the same number of molecules in it as 9/10 of a gram of salt. In other words, imagine if you had 100 glucose molecules and 100 salt molecules. Wouldn't the 100 glucose molecules weigh more because each glucose is a larger molecule? So these two, 5 grams of glucose in 100 milliliters of water or 9/10 of a gram of salt in 100 milliliters of water actually contain the exact same number of solute particles or dots. So those are both isotonic solutions. Now in the second test tube, it shows that this cell, which is still 80% water inside the cell and 20% other stuff, is in this case placed in a 100% water solution. Now a solution that has a lower amount of solute than is found inside the cell is called a hypotonic solution. Hypo means low and tonic means strength. So it's low in solute. It's low in salt, it's low in sugar, it's low in protein, and in fact in this case, it's all water. So it's 100% water. Now obviously if it's 100% water on the outside of the cell and 80% water in the inside of the cell, you should be able to see that there's a concentration gradient for water. There's a higher concentration of water outside than inside. So what is water going to do? It's going to diffuse down its concentration gradient into the cell. Remember, anytime water does diffuse, either in or out of a cell, that's called osmosis. So osmosis is going to occur. And as the water flows into the cell is going to swell up larger and larger and larger, and at a certain point it will burst. And so that, when a cell ruptures or breaks apart, we use that Greek root lysis, which means literally to break apart. So that's not a very happy cell. That's a dead cell. Now, as a mnemonic or memory aid, to remember what a hypotonic solution is, hypo has that very prominent O in the center. It's high in H2O, a lot of water. And as the water flows into the cell will swell up like a hippo. Okay, it's not really the way we spell hippo, but if it helps you visualize what happens when you place a cell in a hypotonic solution, it would swell up. Incidentally, any sugar solution with less sugar than 5 grams of sugar in 100 milliliters of water would be a hypotonic sugar solution, and any salt solution with less than 9/10 of a gram of salt in 100 milliliters of water would be a hypotonic salt or saline solution. Now, the third possibility is to surround the cell in a very concentrated so-called hypertonic solution. Hyper means high, and tonic means strength. It's high in solute. It's got a lot of sugar or a lot of salt or a lot of protein or a combination of all those things, but it's low in water. It's all solute. It's hypertonic, high in solute. Now, in this example we've given here, if the if the hypertonic solution is 80% solute and only 20% water, remember the inside of a cell is normally 80% water. So if it's 80% water on the inside of the cell and only 20% water on the outside, can you see that there is a higher proportion of water actually inside the cell than outside in terms of the proportion of water and solute? So water is going to flow down its concentration gradient, flowing from a higher concentration of water to a lower concentration of water. As the water flows out of the cell, again, that's called osmosis, and as that water flows out, the cell is going to shrink in size. It's going to collapse as the water flows out of it. And as we wrote right here, as the cell starts to collapse and shrivel up, that's commonly called crenation. We say that the cell is becoming crenated. And again, any sugar solution with more sugar in it than 5% glucose, 5 grams of glucose in every 100 milliliters of water, would be a hypertonic glucose or sugar solution. And any salt solution containing more salt than 9/10 of a gram per 100 milliliters of water would be a hypertonic salt or saline solution. Now, in fact, if you were to look at the lab manual on page E7, on E7 are some actual drawings and micro photographs of cells that are actually being exposed to isotonic, hypotonic, and hypertonic solutions. So on page E7, on the left, we have a drawing showing a cell. We are reminded that it's 80% water on the inside of the cell normally, and if the fluid or solution surrounding that cell is similarly 80% water and 20% other stuff, just like inside the cell, that's called an isotonic fluid. And this is a happy cell. You can see it's smiling. And in fact, here is an actual photomicrograph of red blood cells in an isotonic solution, and you can see many of them are smiling as well. In the second drawing, it shows a cell, 80% water inside, that's normal, And in this case, it's surrounded by, I wrote 90% water. It's not 100% water. I see some dots surrounding the cell, some solute, but it's certainly a higher proportion of water outside the cell than what looks like inside. So if it's 90% water on the outside, 80% water on the inside, that means there's a higher proportion of water outside than inside, so water's going to simply flow. Like all chemicals do, it will diffuse from an area of higher concentration to an area of lower concentration. As the water flows into the cell is going to swell up. This picture shows the cell swelling. It's almost at the bursting point. And this is what happens when a cell is placed in a hypotonic solution. And in fact, in the photomicrograph of actual cells on the right, it shows cells, you'll notice they're not smiling, they may even be crying, because they're about ready to burst or undergo lysis as the water continues to flow into them. The third possibility is shown in the bottom figure, the bottom picture. It shows a cell, again, 80% water on the inside, that's normal, and in this case, surrounded by a hypertonic solution, one that is higher in solute particles and lower in water, and in this example, 20% water. So again, what's water going to do? It's going to spontaneously start to flow from an area of higher water concentration to an area of lower, down its concentration gradient. Whenever water flows, either in or in this case out of a cell, that's called osmosis. And as the water flows out of the cell is going to start to collapse, to shrivel up, to undergo crenation. And in fact, here's an actual photomicrograph of red blood cells that do not look happy. They are frowning. They are collapsing as the water flows out of them. All right, so that's the concept of tonicity and how the proportion of water and solutes in the fluid surrounding the cells, how that affects the movement of water, causing water to flow into the cell or causing water to flow out of the cell.